All right, thanks everyone for being here this morning. We'll go ahead and get started. So today I'm pleased to introduce Dr. Swayam Prabha. She's an assistant professor in experimental and clinical pharmacology here at the University of Minnesota. So she obtained bachelor's and master's degrees in pharmacy and pharmaceutics in India before coming to the United States to earn a PhD in pharmaceutical sciences at the University of Nebraska Medical Center. She has an interesting background because after that she went into industry and then has come back to academia. And she's worked on a variety of very interesting and innovative drug molecules and drug delivery approaches, including nanoparticles and organometallic compounds, and what she'll talk about today, which is using cells and delivering nanoparticles, the cellular backpack idea, which is really great. So thank you for being here.
David, can you hear me? Fine. What a day. It's so cold. And I was running around trying to find this CCRV about, I think, 3 or 4 buildings that look alike. That's what actually, if you don't know your path, that's what my seminar is going to talk about. I didn't know the CCRV, but I used, and I was running with my backpack, but we'll be using our cellular backpacks, which know the way to the tumor. And I'm going to talk to you about how we do that with cellular backpacks, targeted tumor drug delivery, which is becoming of keen interest, looking at the patients who undergo severe chemotherapy. As you can see, in deep-seated and metastatic cancers, there has been a poor availability of these chemotherapeutics. And even if we are able to get to the tumors, normal cells also get affected if we give just injectables, chemotherapeutics. Most cancers, we work with lung cancers and ovarian cancers. Most cancers develop these drug resistance because of high-dose chemotherapy, which we are trying to send to the tumor, so the tumor gets killed. But there has been a drug resistance because of high chemotherapy. So it's been becoming of keen interest that we develop a delivery system or encapsulate it into nanoparticle system so that to maximize the delivery in tumors and also to improve the half-life of this drug by reducing degradation and sustained drug release. And I'm not going to talk about nanoparticle delivery system. It's just our mechanism is just to improve the delivery through the nanoparticles. But briefly, over a past decade or 2 decades, there have been many nanodrug delivery systems. Main one being liposomes like Doxil. Doxorubicin has been delivered through liposomes, and also polymer conjugated particles like Abraxane, which is Paclitaxel given for lung cancer. We work with polymeric nanoparticles, and the process by which these nanoparticles have a high drug effect or high targeting to the tumor is because of EPR effect, or enhanced permeation and retention effect. So what happens in a normal tissue, the vasculature is there, but in terms of tumor tissue, when we look at, there are leaky blood vessels. So these large molecules can get into these vessels and hence into the tumor tissue, but there is no drainage. Lymphatic drainage is very minimal, so it gets retained in there. And that's how the delivery through the nanoparticle system is enhanced or retained better at the tumor site. However, soon the nanoparticles were introduced. It came into knowledge that these nanoparticles cannot go further away from these blood vessels. This is a typical example that Rakesh Jain demonstrated with Doxil particles, doxorubicin liposomes, where the red fluorescence is coming from your doxorubicin liposomes. And as you can see, the further in the tumors, it's not, there is no liposomes going in. Hence, we need more and more high-dose chemotherapy to get to the deeper tissues or deeper tumor tissues. Similar effect, we couldn't, even after 10 years or 15 years down the road, we haven't solved that drawback from nanoparticles. You can see this is a tumor section or a breast tumor section where again doxorubicin, fluorescent doxorubicin, is associated at the periphery of the tumor tissues, but not in the deeper regions. So the problem is to get to the deeper regions where the resistance occurs and to kill those tumor tissues. So what happens, and this is what was proposed, is like in a normal tissue, there is a very good tight network of blood vessels where each and every tissue cells is being supported by these blood vessels. However, in a solid tumors or in tumors, there are very few blood vessels which are leaky, but the farther regions, the cells which are resistant cells are very far away from the blood vessels. So that's what happens, that the nanoparticles are taking the path of these blood vessels or getting fluid through the blood vessels, and where the blood vessels are not there, they cannot reach those regions. Another important problem is that the blood supply to these tumor tissues cannot keep up with the tumor growth rate. Tumor is so aggressive, soon they become dense structure, which leads to some solid stress. This was again shown a long time back, but as you can see, from a necrotic region to a normal tissue where the tumors interface with the tissues, there is a variability in the perfusion. And because of this variability in the perfusion, the convection mechanisms or the diffusion mechanism of the drugs are varied. And also in the necrotic region, there is less supply of these drugs. So I talked about a little bit about the blood vessel vasculature or blood vasculature in tumor tissues, which is so varied, and also in the necrotic region, there is less vasculature. There are another mechanism which occurs in these solid tumors. That's the increased interstitial fluid pressure and also the solid stress because of the dense extracellular matrix. That also decreases the diffusivity of drug carriers. So if we look into the tumor tissue, let's look into the normal tissue where the blood vessels are normal and very supported by less dense structure of extracellular matrix. However, in case of tumor tissue, these are very abnormal or either constricted because of high dense extracellular matrix. Some regions it's very constricted, some regions it's very abnormal. And also the fibroblast or what we call as a cancer-associated fibroblast are very high in these tumor tissues. And that leads to, again, stress in the tumor tissue vasculature and vessels constrict. So if a vessel constrict, again, there will be less diffusion of the drug and more passing away of the drug. Drug will not diffuse. So those are the mechanisms which leads to less drug diffusivity. And if you see most tumor tissues, we work with lung carcinomas and breast carcinomas. All these tissues have anywhere interstitial fluid regions about 30 millimeter, anywhere from negative 1 to 30 millimeter Hg. When in normal tissue, it's about negative 3 millimeter Hg. So if the interstitial fluid pressure is increased, there is more stress in the tissues and more diffusivity, less diffusivity of the drugs. That brings us to the problem in these cancers and why we are not able to tackle these cancers that effectively, despite more and more signaling drugs like gemcitabine or other drugs, Paclitaxel drugs, we are not able to tackle this problem because of this intratumoral heterogeneity, which is coming through the solid stress and also through the blood vessel constriction. This leads to chemotherapy-resistant tumor relapse, and also there is an accumulation of different subsets of tumor cells, as I talked about cancer-associated fibroblast, that are hard to get rid of. That leads to different susceptibilities of diverse treatment modalities. Most of my talk will deal with Paclitaxel. That's a first-line chemotherapy used for the lung cancer, and also in ovarian cancer, it's been used once the carboplatin therapy fails. So I'll be dealing with the Paclitaxel quite a lot in my presentation. After coming back from industry, I started my academic career from scratch, basically. I was working in a generic industry for quite a long time, about 9 to 10 years. And I was out of my PhD topic, which was on p53 gene therapy for breast cancer. Coming back to academia, I quickly realized gene therapy is not going anywhere. There are long hurdles to be crossed in gene therapy, and even if gene therapy is going somewhere, the toxicity associated is quite high. I looked into other mechanisms, and obviously biosimilars are picking up, and cellular-based T cells, Keytruda, which came in 2017, T cell-based therapy is picking up. We tried to look into certain carriers which can target these tumor cells. So we looked into mesenchymal stem cells, and it was totally an elevator idea. I'm like, okay, let's see if there is a potential of these cells to carry over drugs. This was our first publication that came in 2014 after 2.5 years of work, and that elevator idea really worked wonders. So, we were able to encapsulate these cells with drugs and also have a good efficacy in tumor cells. And I'm going to walk you some of the data which we are working. Later on, we developed different targeted particles. You can go read. I'm not going to talk about the molecular cancer therapeutics paper, but you can go read. It's about TAT nanoparticles, cell-penetrating peptide nanoparticles, which leads to higher efficacy in lung cancer. So what are mesenchymal stem cells? They have been long in the field since 1974, but mainly used for regenerative therapy. It's a fibroblast-looking cell, a very spindle-shaped looking cell. Cool microscopy. When my high school students work on it, they're amazed by the some of the they have high growth potential, and there are certain markers that are very useful for their targeting to tumors. They are CD34 negative. That's how we distinguish them from normal tumor cells. But the TAR1 and STRO1, they target the extracellular matrix of cancer cells through these receptors. There has been evidence that these mesenchymal stem cells go to the tumor cells. And this is actually mouse emesis, which are labeled with Luke thing and IV injected into a biopsy mice. If you see, there is no tumor in these mice. So within 24 hours, most of the time, the emesis get excreted out of the body because of the exogenous cells. However, when there are tumors, these are Hay tumors in mouse cells, Hay ovarian carcinoma tumors, they stay in the body for even 14 days. And we have even done up to 28 days or even month with a single dose injection, and they stick around in the body for that long. These were just the normal MSCs which were injected, but just loop transfected. And in case of normal mice where there was no tumors, within 7 days, there was a lot of clearance from the body. So that paper and several researchers who have been working on these mesenchymal stem cells, red blood cells, T cells, because of their potential for selective homing to the sites of inflammation and cancer. There is low immunogenicity potential because of MHA class 2 molecule. They are devoid of MHA class 2 molecules. Established biodistribution and toxicology profile has been used in regenerative therapy for years and have been used for generating heart cells, cartilage, bone through a mesogenic process. Have shown positive safety profile in clinical trials data, and also they are easily available from the donors and can easily be reproduced or proliferated outside the body and then re-injected into the body if we need to take it from a cancer donor. There is a possibility of GMP compliant. Coming from industry, I do look into whether it has a commercial potential. Large-scale manufacturing process, and also they can be cryopreserved. However, till 2010, these MSCs have been used as a delivery vehicles by just genetically modifying and injecting the genes, because people are not able to load drugs, small molecule drugs, in these cells. And mainly because they are almost like stem cells. It's a natural repair mechanism of the body. They are like stem cells which overexpress these PGP transporters and hence pump the drug out. So if you try to activate these cells with drugs or culture these cells with drugs, most of the drug is pumped out, or either they just die because of the high dose. There is a poor payload capacity of these cells, and hence most of the people they tried is a genetic modification to target the therapeutic moieties, main one being IL-12. What we looked into is a way to modify these mesenchymal stem cells, and this was the hypothesis that we proposed in 2012, but now we have proved majority of these pathways, with some pathways still not clearly understood to us. What we did, we shielded the drug, like Paclitaxel drug, into these nanoparticles, polymeric nanoparticles, and then incubated the cells with these nanoparticles. These are taken up easily by endocytosis in these cells, and once they carry, we have done studies, and I'm going to show you in few further slides, that these cells become like a small drug factories, because the nanoparticles are there, and nanoparticles are going to release the drug in a sustained manner. We can manufacture it within 4 hours and then inject it into the body intravenously. Once they are in the body, they locate to the tumor. They quickly go to the tumor within 15 to 20 minutes in the tumor, we have shown. And once inside the tumor, once very near the tumor cells, and also it gets to the inert necrotic tissue of the tumors, they release their drug. The mechanism of release of drug in vivo is still not clear to us, whether there is a dose dumping effect. Very few we have seen the dose dumping effect of these nanoparticles, but most of the time it's a sustained release, but we are working on that one. So these nanoparticles, I'm not going to talk about the nanoparticles, but these nanoparticles are formulated using emulsion solvent evaporation technique and has quite a high drug loading. About 148 microgram and in nanometer ranges. So this is the mechanism of how, nanoparticle uptake, that's how we make drug factories. These are little drug factories. Within 4 to 6 hours, the saturation is reached. And you can see it's a dose-dependent effect. As we increase the dose, there is a higher drug loading, but again, we cannot increase too much of the nanoparticles because the polymer itself has some toxicity associated. So we keep about 100 microgram per ml for our in mouse studies, and they form really nice. So green fluorescence that you see is actually nanoparticles with NIR dye or a fluorescence dye. Important thing is when we engineer them or when we load this with these drugs, there should not be loss of migration potential. Migration is important for us because of the tumor tropism the tumor tropism property of this mesenchymal stem cells, and also the differentiation potential. We want these mesenchymal stem cells to not lose their phenotype in a process of engineering. And we found that in vitro, there is no change in the tumor homing potential or migration potential. And also there is no change in either adipogenic or osteogenic differentiation. So that the mesenchymal stem cells do retain their phenotype. They are not changed in the body. In vitro, these are paclitaxel loaded. A549 is a lung carcinoma cell line, lung adenocarcinoma cell line, and MA148 is ovarian carcinoma cell line. Paclitaxel is a little bit more effective in MA148 cell line, which is a sensitive cell line, as opposed to A54 cell line. But we did see that it leads to more than 50% inhibition of cell proliferation at reasonable dose of nano-engineered MSCs. That was in vitro data, but I will not get any R01 grant because of the in vitro data. Nobody gives federal funding if we just demonstrate it in. But we did find a good potential of these nano-engineered mesenchymal stem cells in lung cancer. So take a moment to look at how we did this efficacy study. So this is again adenocarcinoma, lung adenocarcinoma A549 cells, which were injected orthotopically and developed lung tumors. We used control MSCs, which are not nano-engineered, not any modified, because and I will get this question from this crowd. Also, there is some controversial studies that MSCs do lead to tumor growth also. But we used that as a control that there is no tumor growth phenomena observed in these MSCs. Mainly, we use the Paclitaxel solution and also MSCs nano-engineered with Paclitaxel solution. One more thing which we want to talk about is the Paclitaxel solution was given at a clinical dose, which is 120 mg per kg dose administered over a course of treatment, as opposed to MSCs, which has low amount of drug, we could only administer it at 1/10 the dose. However, even at 1/10 the dose, these MSCs were highly effective, and it led to increased survival, about 30 days survival, better than either Paclitaxel solution or even Paclitaxel nanoparticles. Why do we? Moving on, we also did it in a syngeneic mice model because there has been a concern that, okay, if you do an ADMIG-nude mice model, you can get rid of with the Paclitaxel also. What about the immunogenicity associated with this MSC? So we did it in a syngeneic mice model using LL2 tumors, lung tumors. And again, these MSCs were much more effective in LL2 tumors compared to the Paclitaxel solution. We used both mouse MSCs as well as human MSCs, and neither showed any toxicity. So we decided to use human MSCs for our development purpose. So they don't have any immunogenicity potential. This is just the figure where it shows liver toxicity. So one of the side effects of Paclitaxel is when we administered it in tumors or in humans is the neutropenia or leukopenia. It causes, and that's a severe side effect. And as you can see with the Paclitaxel solution, as well as with Paclitaxel nanoparticles, we were able to see some toxicity associated, low white blood cells count. However, in case of MSC, nano-engineered, there were no differences in the WBC count, and other parameters like ALT, AGT remained constant too, so there was no toxicity with this. So we looked closely into the data, and we were trying to find the mechanism associated with the MSC. I hypothesized that MSC do go to the tumors very specifically, but we didn't pretty much know what is going on at a cellular, at a tissue level. So we looked at the biodistribution of these nano-engineered MSCs. And as you can see, obviously this is pretty small. I tried to fit in 4 figures in there, but nano-engineered MSCs had a higher accumulation of the drug. The drug is not degrading because the drug is slowly getting released. But in terms of effect, in case of clearance organs to the tumor tissue, there was 2 to 2.5-fold higher accumulation of drug in the tumor tissue when compared to the Paclitaxel solution. This also brings us to imaging portion, that where the MSCs are located. And if you see, the MSCs are only located in the lungs where there is a tumor. But where there is no tumor, there are no MSCs. So again, very selective accumulation of these cells in lung tumors. And that's what we saw, that rather than in the clearance organ, the drug was present mostly in the tumors, higher accumulation. Whereas in case of Paclitaxel solution, it gets cleared and you will find more in clearance organs. So this talks about the specificity, and looking at the cellular level also, these nano-engineered MSCs were even in the deeper section of the tumor tissues. So they were very homogeneously distributed when compared to the other cells. And this is not the characteristics of just the nano-engineered MSCs. This is also MSCs. If you just put in MSCs and not as a drug factories, they are very homogeneously distributed in these tumor tissues, even in the deeper regions. I talked about that we were able to administer only about 1/10 the dose that was required in the clinical tissue. This was first proof of concept that we demonstrated that, okay, nano-engineered MSCs are able to carry small molecule drugs and are effective, but we can carry only small amount of drug. So we looked into increasing the drug loading in these nano-engineered MSCs so that we can have more of the drug available to the tumor tissues, at least the clinical dose, which is the effective dose. Lot of people work with targeting, what they call as active targeting, where they connect, attach through a ligand like a folate receptor or EGFR ligand. What they do, they attach nanoparticles to that onto the surface. But if you see, these ligands are also present in the normal cells, and it's not very active targeting, but it will go to the normal cells also, these nanoparticles. So we looked into non-natural targets. What we did, we incorporated some azide functional groups in these MSCs, and I'm going to talk about how we did it, followed by the delivery of a very specially modified nanoparticles, creating little satellites in the tumor tissue, and then sending these nanoparticles, which will go click only at the tumor tissues. So once the MSCs are able to find these tumor tissues, we will give the therapy and get rid of the tumor cells. We call it a two-step targeting approach. This not only has very high selectivity, and we were able to increase the loading in these MSCs, but also we could able to reduce the off-target binding of these specially modified nanoparticles, which is important in case of especially cancers like ovarian cancers and also metastatic cancers. So we named it as a glycoengineered MSCs. What we do, we feed these normal MSCs, human mesenchymal stem cells, with something called acetylated sugar. It's a mannose sugar that is used by the body metabolism to express some sialic acid receptors. But what we did, we modified, we used the acetylated mannose, which is a special mannose with azide, so that we can express azide receptors on these body. And then we used a click chemistry to target DBCO nanoparticles into the body. These kind of MSCs can serve both as a diagnostic as well as therapeutic purposes. We can load these DBCO nanoparticles with a drug. And initial studies, we just optimized number of azide groups and also the stable azide groups. Stability is important when we are redosing the nanoparticles, because if the azide receptors are not present in the tumor tissue because of these MSCs, MSCs stick around for about 30 days. But if the azides are not present in these MSCs, our nanoparticles will not click. We found about 20 micromolar concentration of again, I'm not going to go, but since these are non-natural targets, we also wanted to look at the migration potential. Here we are actually changing the receptor profile of these MSCs. We are doing something different to the MSCs. So we wanted to look at the tumor potential. It's not changed. And also, the higher concentration of this sugar is not toxic, but it leads to less proliferation of the cells. And that's why we cut it off at 20 micromolar, which was optimized concentration. Our future work is looking at different sugars to actually have more stable azides. I'm gonna breeze through these things to get to the data, but again, there was no change in the MSC phenotype, which we checked through the differentiation potential. Important was the receptor profile. We are interested in the CD90 or a Thi1 and a STRO1, so that these MSCs can associate with the stromal or extracellular matrix of the tumor. We found that the azide expression on these MSCs, which stays, at least in vitro, we showed that it stays for about a week time. In vivo, it stays much longer. There was no change in the receptor profile, surface receptor profile, also with the azide expression. So this is the data which I was talking about initially. These are MSCs which are glycoengineered with azide receptors, and also the tumors were actually MA148 tumors, which is a peritoneal tumor, and also there is some location in the ovary. It does go to the ovary. We call it an orthotopic ovarian tumor, but mostly the tumor develops in a peritoneum region and also in some ovaries. But you can see there was just single injection of these glycoengineered MSCs. It stays up to 28 days. Obviously, we couldn't take the study longer than 28 days because the mice have started bulging up. A lot of mice, we had to kill it. The tumor is a pretty aggressive tumor. Now, when we cut open in 28 days, there was about 5 times higher the glycoengineered MSCs in tumor tissue as compared to liver. And also there was quite a few MSCs that was going into the ovaries where the tumor was located. That said, what's the potential of these nanoparticles when we inject it? When we inject the MSCs first, MSCs have formed the dipodes. Do the nanoparticles go click there? And that's what we wanted to show in the two-step targeting. So one we did with the intratumoral injection, and I'm sorry, this should be intravenous injection. One we did with the intratumoral injection where MSC, normal MSC or MSC with azide were injected, and you can see there was high accumulation of DBCO nanoparticles in case of MSC azides, as opposed to MSCs, which are normal MSCs, the DBCO nanoparticles do not go click there. This was mainly because of the high clicking and also high retention of these nanoparticles. What is happening when the click chemistry is happening in these MSCs, they go click and it's a covalent linking, the nanoparticles do not go out, whereas in case of normal, when we are normally loading the drug particles, there is a process called exocytosis or transcytosis, which pumps the particles out. So that's how we could able to increase the drug loading in these nanoparticles. We looked at the tumors. So again, this is the orthotopic tumor where we used MSC azides and also MSC azide with a two-step approach where the nanoparticles were injected about an hour after. There was high inhibition of tumor. Even once we took the dose out, then only there was some increase in the tumor. But again, the tumor was completely flat and high incidence of survival in these mice, which were injected with two-step DBCO nanoparticles. When we looked at the localization of these nanoparticles, so to walk through MSCs, azides are actually the one with the azide receptors. These are plain MSCs, no MSCs. Dinosaur is a molecule or an inhibitory molecule that inhibits exocytosis or endocytosis of drug particles or drug molecule in the cells. So we incubated MSC azide with nanoparticles as well as dinosaur, and also MSC with nanoparticle and dinosaur. And you can see that in case of azide nanoparticles, there is a lot more yellow co-localization, which means there are nanoparticles associated with the surface and not inside the cells, as opposed to in case of MSCs, where most of the cells were taken up by endocytosis. But also there was about 10-fold higher loading in case of MSCs with azide receptors. And you can read Advanced Therapeutics paper that just came which talks about where we pre-glycoengineered or pre-nano-engineered, we incubated nanoparticles, and they also show, so it's an alternate pathway. Instead of two-step targeting, we can also inject these MSCs directly into the tumors and get efficacy. This study was done in MA148 tumors, but what we are looking into, why we looked into glycoengineered pre-nano-engineered MSCs is because of glioblastoma. Brain tumors are such tumors where if you inject nanoparticles, they won't go in, but cells can cross that blood-brain barrier. So what we propose to do is pre-glycoengineer or preload it with the drugs and then send it to tumors where it's hard to reach nanoparticles. To summarize, human MSCs incubated with nanoparticles showed a steady increase, and we could manufacture it within 4 hours of the body, 4 hours after taking and culturing, and it doesn't affect the nanoparticle loading in these either the nano-engineered MSCs or the glyco-engineered MSCs, there is no change in differentiation potential as well as the tumor homing potential of these cells. Nano-engineered MSCs demonstrated good inhibition of not only lung tumors, but also ovarian cancer tumors. And we want to make a platform for actually not just cancer specific, because these are not cancer specific. They have a receptor profile that can target variety of cancers. So we want to move the field into other cancers which are hard to reach cancers like brain tumors. So we were able to achieve good efficacy with these MSCs at 1/10 the dose, and this dose sparing effect also led to less toxicity in at least the mice model. Our main goal is to get the therapy to the human, and that's where we are working towards. But in mice, we did see less leukopenia. The newer MSCs, which are glyco-engineered MSCs, they showed selective accumulation in ovarian tumors and less dosing, but higher drug amount. So we don't have to frequently dose these cells in the body. With that acknowledgements, I have quite a few collaborators who I cannot thank enough for initially Tim O'Brien from veterinary medicine. We collaborated with him, and I told you that it was an elevated talk, and as a new faculty, I didn't have that much of startup to even invest into cells, antibodies, and he helped me get those going. Jayant and Dr. Argenta at OBGYN, who is my partner in crime for all the ovarian cancer work. Tim Starr, we have been studying these MSCs in PDX models, and it's been showing good efficacy with Tim Starr. Ramki, who some of you may know, was here and now is enjoying good weathers in Florida. But he also helped me in ovarian cancer work. John Veroha at Mayo Clinic, again, PDX tumors. We have been collaborating quite a few with Northwestern Maciej Lesniak, and he's the person who is working with the brain tumors, and we are collaborating with him. The work wouldn't have been possible without the funding from NIH, and over the years since 2015, I've been lucky and also very lucky to get Minnesota Ovarian Cancer Alliance funding for ovarian cancer work, and they've been very helpful. UIC for all the imaging. Again, I mostly spend my time writing grants, and there are people who work in the lab and help me get this data, help me move these studies forward. Maywan Laek and Saduka, Bud and Tanmoy, they are main in the lung cancer work and the ovarian cancer work. Sushil, Snake, and then my students and also high school students who give me ideas how to make it better and also how to give it to the layman, how to present it to the layman people. The technology has some jargon. Whenever I present it at a Minnesota Ovarian Cancer Alliance, there are a lot of questions. They just say, oh, tell me in a very simple way what your technology is doing. And that's what my high school students help me with. And again, Panem Lab members. With that, I'll take any questions.
So thanks for the very interesting talk, and congratulations on starting from scratch and building up all this cool work. So you mentioned that, endogenous MSCs may be playing a role in response to wounding. And so I wonder if you or anyone's ever looked to see whether there's actually competition between recruitment of endogenous MSCs and the ones you infuse, because maybe you could make the tumor homing better if that competition wasn't there. Just curious.
It's a very good question, and it has been come up several times, and we are trying to look at it. But there was a paper that came in Nature Medicine, I believe in 2011, and they were looking at in case of how the tumor growth potential is. But what happened because of such a aggressive extracellular matrix, whatever endogenous MSCs are recruiting, they get killed. There is not enough buildup to take care of that inflammation.
So the infused ones get killed also?
Infused one gets killed also. That's why we had to do newer, redosing of these MSCs. So that's what is playing the role.
I have just one other question. Well, I have a bunch, but I'll just ask one. So, you hypothesized at the beginning that the MSCs, because they express drug efflux pumps, that is one mechanism by which they're causing the local accumulation of the chemotherapy. And I just wonder if that's been tested explicitly by using MSCs from mice, for example, that have no expression of the efflux pumps. There's triple knockouts for that, so you could see if they're less effective. Because, who knows exactly why this therapy is working, but I'd be curious to know if that's been tested.
It's a good thought, and you should read our JCR paper. We have exactly demonstrated when there are no drug efflux transporters, you can, and nowadays there are like exosomes that is being developed from these MSCs. Yeah. They don't have, so you can load a bunch of drug in those exosomes, but there is an additional processing step for that. So
the experiment has been done with triple knockout.
We didn't do with the triple knockout, but we did it with a PGP knockout.
Okay.
MSCs. We use the inhibitor, and we did see that.
But if you use the inhibitor, it will hit the tumor cells flux pumps too. So I think it's
what we did. It's a in vitro experiment, because what is happening is so I think we can do that experiment also. We did just the in vitro where only the population was MSC population.
Hi, great work. I just have two questions. One, you mentioned earlier that some of the MSCs could potentially induce tumor growth, and the ones that you guys used didn't. So what's the difference of the MSCs that you guys are using? And number two, what is the metabolism of this mannose coated Azides, or I forgot? How do you feed that to the cells, and how long does it take to actually express those markers for better drug delivery? Thank you.
Good question. And answering your first question, can you repeat?
So what was the difference between because you mentioned the MSC in some places can promote tumor, and so in your case it didn't. So what was the difference in like the underlying cellular biological process?
It's a roundabout answer, because I don't have answer for that question. But what we think is, like, since we are able to engineer them with the loaded drug particles, we are not injecting high number of MSCs. We do see, like, if you inject MSCs at a higher numbers, those papers do inject MSCs at much higher numbers. So when there is higher, how I would say, like, more killer cells, that balance, if it is maintained, what we are trying to do is maintain that balance so that there is no tumor growth potential. But if the MSCs start accumulating and differentiating, they will start forming small tumor spheroids, and they do use MSCs for tumor spheroids. So it's the balance between the number and the killing of the cells, so it doesn't cause tumor growth. But again, if we inject higher amount, we would start seeing, and those were the reports. There is another report because they are trying to when they are genetically modifying, they are trying to do CXCR4 signals. So that's again causing a little bit higher tumor growth. It's like a bystander effect they talk about. That's what we think. But again, it's not a positive answer. Like I cannot say with confidence that this is what is happening over here. In case of your second question again.
Metabolism of the mannose-coated azide.
So
as a
mannose
process.
sugar, what we, and I think I had, and this is a very chemistry slide. But what happens, we incubate it with about 3 days. It takes about 3 days for that metabolism process cycle to happen in case of mannose. And as I mentioned, I'm trying to work with the different sugars, which are high metabolism sugars like glucose and galactose. They take a little bit less time to express azides. And that's what we are trying to play with, is to minimize the time and stability of these. So obviously mannose is it's a simple sugar, but its metabolism path is quite high. So it takes about 3 days in vitro to culture these, express it with azide receptors. And also those azide receptors are not, since these are non-natural receptors, they are not that stable. We see 60% decline by the end of the 7 days. But what we do is, like, as soon as we have, like in 3 days, we have maximum receptor, we inject it in the mice. It doesn't decline that rapidly.
Great concept. Thank you for telling us about it. I just want to clarify to make sure. So this is kind of a ovarian cancer alliance question. So you are pre-treating the MSCs with the azide coupled mannose to create the MSCs that have the mannose? Or are you trying to put the mannose on the tumors?
That have azides.
The dyes. And then once they're in and in position, you're going to go ahead and inject the mannose targeted nanoparticle.
Azide targeted nanoparticles.
Okay, the azide. Okay.
Yeah, the mannose gets metabolized. It's the azide portion that's remain. So if I can zoom this, so this azide portion stays on the cell surface, if you see. The mannose gets metabolized. So this is the receptor which we are targeting our DBCO moiety with, and this is the sialic acid that gets converted. The mannose gets metabolized in the cycle, in that Krebs cycle.
Okay.
And
all right.
Other questions or comments? Okay, let's thank her again.